Welcome to NPTEL online certification course on Pharmacognosy and Metabolic Engineering. This is lecture number 56, where under the broad domain of phenolics, I am going to cover alcohol acetyl transferases and volatile phenolics. Let us go to the concept to be covered. So, we will cover about the concept of alcohol acetyl transferase, what sort of enzyme it is, how it catalyzes the reaction, and then it belongs to BAHD family. Why it is called BAHD family, we will see. What are the other members of BAHD family, what is their function, we will see. And then we will see that is one such alcohol acetyl transferase genes, if such gene can be transferred from a source plant to a target plant, what effect it will make on the volatile production. And then finally, we will talk about the biosynthesis of eugenol and chavicol, which again the ultimate step of this actually it comes under alcohol acetyl transferase only. So, this will be entirely board work. So, let us go to the board now. So, overview of phenyl propanide metabolism, I always show this slide in the beginning. So, this is not also an exception. So, basically the so C6C3 compounds. So, we will be dealing with the C6C3 compounds only. So, that leads to the formation of different compounds like eugenol or chavicol. And when we will talk about the volatile ester components, there C6C1 components will also come. So, the purpose is that is all phenyl propanide metabolism. So, when you will talk about the volatile phenolics, it will come to your mind is one is the eugenol. So, eugenol is produced in so many plant, the most important source is clove, whereas isoeugenol, which is produced from the synthesized in the flower of ylang ylang. Similarly, thymol is from thyme, carbapenem is from origanum. So, all these contains this phenyl ring. So, it's all are phenols. So, with different names, it has all different names, and they have specific physical properties, specific gravity and all these things. So, these are required when one try to isolate the essential oils from the plant, and then after purification, what is the quality of the oil, then such activities like specific gravity, refractive index, density, all these are to be checked. So, anyway, that is not our concern. Let us now go to the concept of alcohol acetyl transferase. Or AAT. So, first there should be one alcohol, say ROH, that will join with a acetyl CoA, and then if the reaction is catalyzed by alcohol acetyl transferase, then it will make acetate plus coenzyme. Once again, this is alcohol, this is acetyl coenzyme A, this is acetate, and this is CoA. So, the acetate is basically the ester. Okay, it's now let me give you some example. Okay, for example, I will start with a monoterpene examples. So, which you are well aware. So, one such example is geraniol. So, geraniol is found in rose. Geraniol, by the action of alcohol acetyl transferase, it makes geranyl acetate. Okay. So, of course, in each case acetyl CoA is required. So, which I am not showing here. Similarly, another nice example will be cis 3 hexenyl alcohol. Cis 3 hexenyl alcohol are usually found in grass, and when grasses are cut off by using manually or by machine, you will see a typical aroma cover the air. So, that is nothing but this cis 3 hexenyl acetate. So, upon this wounding, the alcohol acetyl transferase comes under the exposure of air. So, that actually enhances the reaction, and acetyl CoA is there in the cell. So, it makes 6,3 hexenyl acetate. So, all these are volatile compounds. So, volatile compounds means they emit. So, another example is 2-phenyl ethyl alcohol, say 2-phenyl ethyl alcohol. This, by the action of an alcohol acetyl transferase, it makes phenyl ethyl acetate. Phenyl ethyl acetate. Again, this is a volatile compound. So, all these are volatile esters. Okay. Now, like this, several alcohol acetyl transferase have been characterized. So, then once these enzyme properties were studied, scientists decided to put them under a specific family, and the family they have named as BAHD family. BAHD family, which covers plant specific alcohol acetyl transferase. Now, why BAHD? So, that we will see. So, BAHD, B comes from BEAT. From this, B goes AHCT, then HCBT, and then DAT. Okay. So, in the next slide I will write the full name. So, this is basically the sequence of discovery of plant AAT enzymes. So, plant alcohol acetyl transferase first enzyme discovered was BAT, next one was discovered AHCT, then HCBT, then DAT. Now, there are more than 30 or even more alcohol acetyl transferases, but they have given the name BAHD based on the first 4 enzymes discovered, taking the first alphabet from each. So, this is just the history. Now, let us see what is BEAT. So, our point of discussion is alcohol acetyl transferase, the BEAT. BEAT stands for benzyl alcohol acetyl transferase. So, this was discovered in a plant called Clarkia brewery. CLARKIA, this is K. Clarkia brewery. The next one is AHCT, anthocyanin O hydroxycinamide transferase. So, this is discovered from Gentiana triflora. The third one is HCBT, which is N hydroxycinamyl benzoyl transferase. This was discovered from Dianthus caryophyllus, CARYOPHYLLUS. And the fourth one, I hope you can remember this because I have covered this in the alkaloid chapter. This is diacetyl vindoline 4O acetyl transferase. So, that was discovered from Catharanthus roseus, leading to the formation of the monomeric indole alkaloid, which is vindoline. So, the product of BEAT is basically benzyl acetate. The product of BEAT is benzyl acetate, the product of DAT is basically the vindoline. Anyway, now, so I have mentioned about this that geraniol. So, geraniol is a common volatile substances which is found in the rose, and geraniol can be converted into geranyl acetate. So, if you smell the rose petal, you will find there are few rose cultivars which are more pungent in smell, and there are few cultivars which are more fruity in smell. So, the more pungent is basically that is because of the geraniol, that is because of the phenyl ethanol. So, if we write rose here, rose fragrance. So, here it consists of two components, one is the alcohol components, another is the ester components. So, in the alcohol components, these are geraniol, phenyl ethanol, and the ester components are geranyl acetate and phenyl ethyl acetate. So, what I said, alcohol are more pungent in smell, whereas ester components are more fruity in smell, and you can distinguish different rose with this, because the rose, what I found, the rose with the lighter petals, okay, lighter red color or more towards pinkish, which gives a more of fruity smell, but the rose with the more deep, which you will find more of the geraniol and phenyl ethanol. Okay. So, it abundantly consists of this alcohol acetyl transferases. So, that means the rose scientific name of rose is Rosa hybrida. Therefore, RHAAT, we can write RHAAT, that is the rose specific alcohol acetyl transferase, which converts geraniol to geranyl acetate and phenyl ethanol to phenyl ethyl acetate. And now, in this context, let me tell one simple but interesting metabolic engineering applications. So, I go to the next slide. Alcohol acetyl transferase metabolic engineering applications. Okay. So, what I was talking about? I was talking about RHAAT. So, for the RHAAT enzyme, the substrates were geraniol and phenyl ethanol. So, these are the substrates. And these are present in the rose petal. Now, so this is the source gene. Now, the target plant was chosen as petunia. As Petunia hybrida. Why? Because those two above substrates, both geraniol and phenyl ethanol, these are present in petunia. So, that is important, because by now it is clear, if you want to do metabolic engineering, you should be sure that the ultimately the substrates should be present in the target plant. Otherwise, gene can be transferred and expressed, but it will not do the proper function. So, so they have taken Petunia hybrida CV Mitchell, that is one, and they have introduced this RH AAT under the control of a promoter and a terminator. So, here they have used a constitutive promoters, that is CAMV35S. You know that I have mentioned this is a constitutive promoter, that is it can be expressed throughout the plant, and this is basically a NOS terminator was used where the transcription will terminate. Okay, and this along with an antibiotic resistance marker was put it in the vector, and the gene was transferred through agrobacterium mediated transformation to Petunia. So, Petunia is well standardized plant, gene transferred to Petunia. Petunia hybrida CV Mitchell, CV means cultivar Mitchell. So, now the transgenic plants, now you have the transgenic Petunia hybrida plant expressing which one? RH AAT gene of Rosa. So, what they found that transgenic Petunia flowers emit higher level of. So, we go here, flowers basically they emit higher level of benzyl acetate and phenylethyl acetate. Benzyl acetate and phenylethyl acetate. So, what it indicates? It indicates that in planta availability of substrates was properly utilized by the transgenic plant where RHAAT gene was put, and as a result of that it produces the desired product which was expected. Okay. Now, I have also mentioned that this rose AAT also has the substrate specificity for geraniol, because it converts geraniol to geraniol acetate. However, no geraniol acetate was detected because the Petunia is not producing any geraniol. But what they have done, and what they also found, that the preferred substrate for RHAAT inside the Petunia plant is for the benzyl alcohol than phenylethanol. Okay. What I mentioned here, why benzyl acetate is formed? Because let me little bit explain. Because this Petunia flower contains normally benzyl alcohol in higher amount, than phenyl ethanol. That is why the emission of benzyl acetate was much more than that of the phenyl acetate, that is clear. Now, geraniol acetate is not produced because no geraniol was available, but what now they have done, in order to test the function, they have done feeding of this transgenic tissue with geraniol, or even they have used octanol, and that leads to the production of respective octanyl acetate and geraniol acetate in the transgenic tissue. So, which indicate that volatile production was possible depends on the substrate availability. That means, when you fed in vitro substrate into the petal tissue in transgenic say petal tissue, respective acetate production occurred. Okay. So, this is all about this work, and this beautiful paper, but simple, published in the famous one of the famous journal, which is called Plant Molecular Biology. By Israel group and collaboration with Purdue, and this was published in the year of 2006, volume 60, page number 555 to 563. So, the interested students can, if they can get the paper, they can read a little bit more, but the major essence I have given. So, with this now I will move into the other aspects of volatile esters. Before I move into the eugenol, I will also briefly mention about this rose fragrance. So, although I have mentioned phenyl acetate, phenyl ethanol, but how this is formed we need to see quickly, that is the volatile fragrance in rose. So, it all started with phenylalanine, and this phenylalanine converted into 2-phenyl acetaldehyde, I am not drawing the structure because of shortage of time, but in the next class structures will be shown. So, then this phenyl acetaldehyde will be converted into 2-phenyl ethanol or phenyl ethyl alcohol, and this will be converted into 2-phenyl acetate, and also from phenylalanine phenyl pyruvate can be formed, and which can also contribute to the formation of phenyl acetaldehyde. So, this is 2-phenyl acetate, 2-phenyl ethyl acetate, 2-phenyl ethyl acetate, and this is phenyl pyruvate. So, the enzymes are PAAS, phenyl acetaldehyde synthase, then phenyl acetaldehyde reductase, then alcohol acetyl transferase, and here also aromatic amino acid acetyl transferase, and this is PPDC, phenyl pyruvate decarboxylase. So, this is more or less in brief the volatile fragrance pathway operating in rose leading to the formation of phenyl ethyl acetate. Okay. And this phenyl ethanol can also be stored temporarily as phenyl ethanol, as phenyl ethyl glucoside, and depends on necessity it will be broken down. So, now we will talk briefly about the biosynthesis of eugenol. Eugenol and chavicol. So, this all started with either coumaroyl alcohol or coniferyl alcohol. So, I will only draw the structure at towards the end. So, the basic structures I am not drawing as I have drawn earlier. So, para coumaroyl alcohol or coniferyl alcohol, let us say para coumaroyl alcohol or coniferyl alcohol, these are the monolignals. So, these two can join with acetyl CoA. That means, either this joins with acetyl CoA or this joins with acetyl CoA, and it makes respective para coumaroyl acetate coumaroyl acetate or coniferyl acetate. So, one such structure will be like this. This is coniferyl acetate. In the next step, coumaroyl acetate and coniferyl acetate will carry out different reaction. For example, the coumaroyl acetate will be utilized for the production of chaviol or eugenol. The enzyme here is, the first one is called the enzyme which joins CAAT for here, CAAT, coniferyl alcohol acetyl transferase, and this one is called CFAT, coniferyl alcohol acetyl transferase, and CAAT stands for coumaroyl alcohol acetyl transferase. Similar sort of enzymes. Okay, you should not get confused. So, I just change a little bit this drawing here. So, I put it here. CAAT. Okay. So, the next one is called EGS or eugenol synthase. So, eugenol synthase, it can produce both the things. So, R equal to H, R equal to OCH3. When R equal to H, so it produces chavicol. So, with cumaryl 1, it produces chavicol, CHAVICOL, whereas with coniferyl it produces eugenol. Similarly, for isoeugenol synthase, which is called IGS. When it uses cumaryl acetate as a substrate, it produces a product which is called T-anol, trans-anol, whereas when it used coniferyl acetate as a substrate, it produces isoeugenol. Okay. Now, chavicol subsequently converted into methyl chavicol, eugenol as methyl eugenol. So, or and similarly, the anol will be converted to anethole, ANETHOLE, whereas isoeugenol will be converted into methyl isoeugenol. So, the enzymes responsible for these reactions are either CVOMT, chavicol O-methyl transferase, CVOMT, or eugenol O-methyl transferase, which produces methyl eugenol. Here also the similar enzymes also function like CVOMT or EVOMT, it makes respective products. So, and this is called, or it is also called IEMT, and this is CVOMT. Okay. Eugenol and isoeugenol, the structure slightly differs. So, I just draw it here, a structure of eugenol. So, in case of eugenol, this is the structure of eugenol, and if it is isoeugenol, then same structure, only this becomes this. So, remaining things are same. So, this will be isoeugenol, and in case of chavicol it is just H, no OCH3 will be there, because chavicol originates from the cumaryl, and whereas the eugenol originates from the coniferyl. This is only the difference, and in the most simplified way I try to explain. So, the important point is this: in order to produce eugenol, the first enzyme is coniferyl alcohol acetyl transferase. Again it is a BAHD family, because it requires coniferyl alcohol and acetyl CoA. So, acetyl CoA is required every cases. Okay, I have added here. Yeah. So, it is also, it belongs to BAHD superfamily, BAHD family enzymes. Similarly, CAAT requires cumaryl alcohol and acetyl CoA. So, this is also a BAHD family enzymes, and the next one is basically eugenol synthase or IGS, isoeugenol synthase. So, and that leads to the formation of the respective products, and then the methyl transferase will come and work, and that leads to the formation of final the methylated product of eugenol, which is methyl eugenol or methyl chavicol, methyl isoeugenol or anethol. So, these are all the volatile phenolics, and it plays important role, particularly defense, but some of these compounds are useful for our healthcare benefit. This is in brief more or less the okay. But in addition to that, there are one more aspect I will just finish before I end this class. I have mentioned about the so this is again a aromatic alcohol acetyl transferase. So, for example, the structure benzyl alcohol. So, what I have said, BEAT uses acetyl CoA and benzyl alcohol, but BEBT or BPBT uses benzyl CoA plus benzyl alcohol, and that indeed joins by the enzyme called BPBT, earlier it was called BEBT, and which makes the product which is called benzyl benzoate. So, benzyl benzoate are important components of floral fragrance. Similarly, phenyl ethyl alcohol or phenyl ethanol can also joins with benzoyl CoA and makes phenyl ethyl benzoate. Phenyl ethyl benzoate. And the enzyme is basically same, which is BPBT or BEBT. So, these structures I am going to show you in the next class. So, this is with this I will rather end the class. So, what I have mentioned here, from chavicol to methyl chavicol, I have mentioned about this. So, this is the monolignol pathway, you see CAD and CCR. Similarly, coniferyl alcohol, then it produces eugenol and then methyl eugenol. Okay. And so, this is again the same CAAT, and then it produces eugenol, Chabicol synthase, whatever, it is all same. So, only the way of different representation. So, with this I end this class. Thank you.